I'll give a quick intro because I'm sure many of you know Dr. Lundgren. It's the reason maybe you're tuning in today. So he doesn't need too much of an introduction, but he's going to be talking today about we've had kind of expanding on our first two webinars. This will be the finale of our Neonic webinars in this series. The first two speakers, Dr. Bredesen and Dr. Wu Smart, did an excellent job of really introducing us to what neonics are, how they're moving through the environment, and some of the concerns of or unintended consequences of those insecticides on our insect friends. So, Dr. Lundgren is going to be taking that a step further and kind of discussing what they're doing in higher level organisms and how they're affecting maybe society at large. So, without further ado, I will turn the reins over to Dr. Lundgren and remember to put any questions you have in the Q&A. So take it away, Dr. Lundgren.
All right. Well, thank you very much. Yeah, we're gonna cover a lot of ground today. We're going to be talking a little bit about some of our newest research that investigates how neonicotinoids are influencing wildlife species. But also, I kind of wanted to go a little bit deeper than that and kind of think about how risk assessments happen, some of the constraints and or strengths and weaknesses of risk assessments, to kind of contextualize not just this talk, but kind of all of the talks that we've been going through right now in this series. So, yeah, for those that don't know, yeah, I run the Joint Ichthyosis Foundation, and also Blue Dasher Farm. If you don't see me looking at the camera, it's because I'm looking at the local audience here. So yeah, not trying to kind of throw you off or anything like that. But without further ado, um, Paul Herman Mueller, he's a happy gent. There he is, right there on the screen. He's happy for a pretty good reason. Back in 1948, he won the Nobel Prize for medicine. What he was studying that got him that Nobel Prize was pretty influential, one could say. What he was interested in, what he did that was so monumental was that he discovered a chemical. The chemical it lasted a long time. It was really cheap to produce, and it was blindingly effective at killing insects. And that's really important because back at this stage of human history, the battle between humans and insects had already been waged and won by the bugs. And so what Mueller created in the form of a synthetic insecticide suddenly tipped the balance between this ecological war that was going on between humans and insects for millennia, millions of years. So yeah, he created this chemical and we used it. It was great, right? Because at that time we started putting it on things like our soldiers, right? Because insects have killed more soldiers than any bullets or bombs have killed. Through disease transmission, they have literally turned the tides of war. And so the folks that had insecticides, their soldiers ended up surviving so much better than anybody else out there. This was a huge, huge advance. We started using it in our houses, right? Because insects ended up being a major pest of human structures as well as consuming so much of the food that we required. I think 30% of all grain crops ended up being lost to insects at this point in human history. And we started to play it out and put it out into wetlands. And I can move this out of the way a little bit. Wetlands and other natural areas. There was actually a WHO, World Health Organization, worldwide campaign to eradicate mosquitoes off of the face of the earth. It didn't work. In case you didn't notice, that did not work. But what it did was suddenly we had these chemistries that we could apply to the environment that were supposed to be very safe, according to all of our understandings of the day, to humans and to animals with backbones. What was that chemical? Well, you kind of see it right there, right? In one of those pictures, DDT. And we hear about that a lot. This was the poster child of a lot of these early chemistries that had been developed, organochlorines and organophosphates. Um, and and so we started to think about this because they were such a powerful tool. We started to think about these chemistries as a prophylactic, as an insurance policy. A lot of times we didn't even have to know whether or not the pest was out there. We should just apply the insecticide. It's cheap and it's easy to use. Okay. The problem was that, and it took decades to understand this, but there was unintended consequences of these synthetic chemistries. They started to accumulate within food webs. When I was growing up, it was like an event. We stopped the car, kind of an event, when we'd see a hawk or an eagle. And that was because these early chemistries would bioaccumulate within food webs and they'd manifest themselves in these apex predators. And in the case of hawks and eagles, it would weaken the eggshells of those birds, such that when the mothers went to roost on their nests or their clutch of eggs, they would literally crush their offspring. And so we almost killed off the American bald eagle using DDT and these early organochlorines. We still find DDT in human breast milk and throughout food chains, in fish and in other things, other animals out there. So the longevity of these early chemistries was really profound. There were more problems because it wasn't just wildlife that was starting to show symptoms of these early chemistries. No flies on me thanks to DDT. Let's put this stuff on our babies, right? Um, there was all kinds of things, birth defects, breast cancer, diabetes, these neurological issues, because many of these insecticides, both then and today, a lot of these insecticides are neurotoxins. That's their mode of action. And you guys learned about that in Mike Bredeson's presentation the other day. To add insult to injury, so we see these declines in wildlife. We see these declines in human health, things that we'd never experienced before. Adding salt in the wound was that the insects started to chew the insecticides up and spit them back at us again. They became resistant. So all of these target pests that we were trying so hard to control, things like mosquitoes, things like spider mites and California citrus, all of these where these were ground zero. These were some of the first targets of a lot of these insecticides. They became resistant and the insecticides were no longer effective against them. So this was rolling along throughout history, right? The first 10, 20 years of the early history of these synthetic chemistries. And seeing the problems, there was a bunch of hippie scientists out in the University of California, Stern et al., and they came up with a really fundamental idea, a major sea change in how we interface with both pesticides as well as the natural world around us. They came up with this thing called the integrated control concept, which is now known as integrated pest management. What they said that was so unique was that killing every insect was a bad business decision. They could turn this all into economic currencies. That actually most of the insects out there we actually need, we can't live without. That was really pivotal. Um, because until then, with these early chemistry uses and using them and thinking of them as a prophylactic, they were so cheap that it just always justified their use. But the reality was is that there was a lot of hidden costs, economic costs, profitability costs that weren't being reflected in those early observations. The other thing that these guys said that was really influential was that they said that you shouldn't be using pesticide as the first line of defense against pests. It should be the last line of defense against these pests. That there were other natural processes that were much more effective and long lived. Things like using natural enemies, predators and parasitoids, and things like this that eat the pests that you could select for plants that had natural resistance to these pests, and that there were things like delaying planting dates or crop rotations or other cultural practices that could really influence pest populations far more effectively than using a chemical spray. And only when all of those other aspects, all of those other tools in our tool belt failed, that's when you would apply these pesticides. Simultaneously to when Sternatol were coming up with this concept of integrated pest management, there was another movement that was afoot, and it was really spurred on by a pretty special lady named Rachel Carson. Many of us have heard of Silent Spring. Rachel died in 1964. She never actually got to see the outcomes of what it is that she did. What made Silent Spring extremely important, Rachel Carson was a wonderful storyteller. She was able to help people connect. She was observing all of these problems in the environment, right? She was seeing things that happened out that these species were in decline, and she could connect the dots. And she was able to articulate the that in Silent Spring in a way that never really had happened before. And it raised these issues and it made them personal and something that people, the general public as well as policymakers could connect to. And the other thing is that she kind of really was an emblem of was the policymakers that were developing or saying that these pesticides were safe and okay. They were being bamboozled. That industry had a concerted campaign to convince them with misinformation that these pesticides were safe and that there were no problems. Silent Spring called that one out too. The outcomes of that book was the United States Environmental Protection Agency was formed and major legislation was enacted that sort of changed our understanding of pesticides in a lot of ways. Pointing out here, this was not a partisan issue. Nixon was the one who came up with the EPA, okay? This was everybody was affected by the degradation of our natural areas, right? Everybody was affected. They could all tell stories about human health problems that they were experiencing. And so this made a lot of sense. The way that they got this through was pretty brilliant. And it lasted the effects of the US EPA. I mean, realistically, they had about a 3 to 5 year window of effectiveness before agro industry figured it out and completely steamrolled the EPA, pulled any teeth out of anything that they were trying to do. But in those 3 years, they enacted legislation and managed to get DDT and all organochlorines banned. They managed to major steps forward, the likes of which we've never seen again. So between this whole interface, all of this stuff is culminating, right, in the early 70s and early 80s, right? There's this sweet spot of risk assessment. And it really sort of forced our hands of, okay, so these pesticides, we need to be using them with our brains on, right? We need to have our thinking caps on when we're thinking about pesticide use because it's not all roses. Number two, there's actual risk to humans as well as to the environment that are really challenging to predict. How do we go about that process? The Red Book of the US EPA is still the Bible of this process, and it formalized what risk assessments look like. It formalized how we conduct these things. Essentially, risk is really easy fundamentally to assess, right? It's two elements, hazard and exposure. And we'll talk about those things. So, hazard, this is a great example of a hazard assessment. It's one of these that one of me and one of my students did before it was cool. We were looking at dicamba and 2,4-D and their effects on beneficial predatory beetles. This happens to be one of my favorite bugs, by the way. It's Poliomygilla maculata. So this is okay. So hazard assessment's really important. You have to figure out what is it that we're really trying to test. This is the toxicity side of things. Okay, so are we worried that the hazard is death? Is it reduction in reproductive capabilities? Is it population growth? Is it something else? But we have to be very clear in that formulation of what question it is that we're asking. And then you have to do a very complex series of experiments in order to try to measure that. So those aren't easy things to do. All right, so that's how you get to a hazard. Then you have to think about exposure because the most hazardous chemical on the face of the earth amounts to zero risk if the organism that you're worried about is never exposed to it. So exposure is an equally challenging, sometimes more challenging approach to trying to understand and characterize risk. So in this case, we're looking at things like neonicotinoids, and I'll talk about them in 1 second. I'm kind of jumping the gun a little bit, but you kind of get the idea. If we're worried about honeybees being affected by seed treatments in cornfields, how on earth would they ever be exposed to those things? Well, they could be exposed to the seed dust during planting. They could be exposed to things like guttation fluid. Most people don't know it, but corn plants bleed water out of their leaves, and a lot of insects and other organisms rely on that water. As if you've ever walked through like a cornfield in the morning, some of that's due, but a lot of it is guttation fluid. The plant is kind of oozing out all of this water from their phloem to try to regulate osmotic pressure. Happens to have a lot of neonics in it. And then other ways would be things like pollen or something. And those are just the direct routes. So you have to try to understand all of the different ways that a pesticide might get through a food web to affect your organism. That requires a lot of really in-depth understanding of the nutritional ecology and trophic placement of organisms. And then once you do all of those experiments, right? You've characterized the hazard. You've looked at all of the different exposure routes. Then we have to decide how many dead lady beetles is okay. And that requires societal values at that particular time. It also requires understanding of the complexities of the natural world. So, needless to say, risk assessment is really challenging. Add to this a few things. So when you go down to the hardware store or wherever, and you walk down the aisle because you got, I don't know, something you want to kill, and so you buy a jug, and you read on the side of the jug, and it's like there's an assumption there, right? There's an assumption that it's safe, that somebody's watching, and that there's been a risk assessment done on it, and that we know the implications. of its use. I've been studying risk assessment for 25 years. I've advised the US EPA and how to regulate it, European Food Safety Authority, right? I can honestly say, after all of that experience, nobody's watching. Nobody is watching. How on earth could they? We have 30,000 different formulated pesticides that are currently regulated in the United States, 570 active ingredients alone. Plus, every time that you put one of those active ingredients into one of these formulations, it completely changes the risk scenario. So that we've got tens of thousands of potential risk assessments that have to be run. Okay, then you have to try to do all these hazard and exposure experiments on the thousands of potential species, non-target species that occur within the environment. No way. And then looking at the lethal and sublethal effects under a whole bunch of different climatic and environmental scenarios. Who is going to do that work? I mean, each of these risk assessments takes years to conduct. The reality is that the only people that are providing regulatory data on the safety of these pesticides are the chemical registrants. The industry that's actually creating the product is telling us how safe it is. And the US EPA just goes through and checks the boxes at this point. There's very few scientists. Judy Wu Smart happens to be one of the only ones left studying the effects of pesticides on bees. Fast forward, all right? So this is all going on, right? All of this is happening. We're starting to understand the consequences of prophylactic use of pesticides on the environment and human health. Risk assessment is evolving in order to be able to assist us in this process. And suddenly we wind up in the early 2000s. And a sea change has happened in our understanding or use and application of pesticides. Chemical seed treatments are suddenly, these are soybean seeds, right? They're not grown that way. They're treated, all of these different colors of the rainbow, right? They're treated with different chemistries. Neonicotinoids happens to be one of those. They're systemic. So they're meant to protect these seedlings from early season herbivory, especially. Simultaneously, there's the development of GMOs, genetically modified organisms that express Bt toxins and other pesticides within the plant itself. The genome is actually altered. This was a major step in the evolution of pest management, because what it did is these products suddenly became marketed as prophylactics, as an insurance policy against pests. You don't even have to know whether the pest is in your field. For a minimal fee, you can buy this thing and just know that it's going to be killed. You're not going to confront that issue anymore. Sounds familiar, doesn't it? The exact same scenario that we had forgotten happened in the 1950s, we're reliving today. And the consequences we're reliving today. So why have we, boy, it took a long time before we even started talking about neonics. But I think that this is really important and fundamental to understanding where we are and why we're confronting some of these issues. So why are neonicotinoids so important? Why on earth would we be focusing 3 hours, 4 hours, probably with all of the questions and stuff like that, 4 hours of time just on one class of insecticides? I'm not going to go into the mode of action and all of that stuff. Needless to say is that approximately 11% of the terrestrial land surface of our country right now is treated with neonicotinoids. One corn seed, like you see up here, you see a whole bunch of corn seeds right now. One corn seed has enough insecticide to kill 164,000 bees. That's incredible. the toxicity, the effectiveness of these. These are 5,000 times more toxic to insects than DDT was. That's a big step. And they're marketed as seed treatments, and that this is a way of reducing the actual amount of active ingredient in the environment. And that's true. But what's ignored is the fact that the active ingredient is so much more toxic than anything we've ever seen as humans before. So, what are the implications of these things? You guys have heard some of the things from Judy and from Mike on insects and how they're getting into the environment. One of our friends out in Montana, this was a number of years ago, one of our friends out in Montana called and she said that we were experiencing or that they were experiencing in her county declines in the white-tailed deer population. That 70% of these deer ended up having these weird jaw deformities. And the males, about 70% of the male deer ended up having these genital deformities. And she wanted to know whether or not neonicotinoids were causing or related to some of these declines in the health of the deer. So we got a graduate student. Her name was Elise, and she went to South Dakota State University. And this project was in partnership with the wildlife department at SDSU. And she conducted a study that will never ever be repeated again because it was so incredibly difficult. She took captive white-tailed deer and she force-fed them imidacloprid, which is the neonicotinoid, in their water over a couple of years. And we watched the effects of the neonicotinoid on these deer. What did she find? Number one is that the dead fawns ended up having substantially more neonicotinoids in their, or imidacloprid within their spleens. And Elise tested all kinds of different tissues, okay? She tested brains and blood and milk. Milking a deer, that's not easy. Liver, spleens, okay? All kinds of different tissues. The quantity that was found within the spleens was highly correlated with fitness effects. And so that became sort of this magic organ that we ended up focusing on for white-tailed deer as well as other organisms. The more imidacloprid that we were finding within the spleens of these deer, it was associated with reduced fawn body weights. They were smaller. Understandably, they also had smaller organs. Okay. So we're seeing these insecticides seem to be associated with smaller animals. Reduced thyroid hormone levels within the fawns. The fawn jawbone lengths were being affected. It sounds pretty familiar. And the activities in the adult deer seemed to be significantly affected the more imidacloprid that they had within their spleens. Boy, the thyroid hormone levels really hits home. I mean, how many people in your family has hyperthyroidism right now or hypothyroidism right now? It's crazy. There's a few members of our staff that do. What's the activity level in the adult deer? What's a slow deer in the wild? It's a dead deer, isn't it? So these insecticides that were never meant to have effects on higher organisms seem to be having significant health implications for white-tailed deer. Okay. That's the hazard assessment, right? You force-fed those deer imidacloprid in their water. Of course you're going to see an effect. What kind of an exposure do we see out in the field? So this is what we were able to generate under laboratory conditions. This was the amount of imidacloprid that we could get into those deer. All right. North Dakota ended up giving us a whole bunch of deer spleens from hunter-killed animals. And they gave it to us in order to test this to see what kinds of levels we were seeing out in field conditions. Those white-tailed deer under natural conditions had 3 times the level that we were able to experimentally generate under controlled conditions. What on earth? These things, these pesticides, these neonics are getting into wildlife at levels that we would never have predicted. We're not that different than a white-tailed deer, are we? How about a cow? How much different is a cow from a white-tailed deer? Not very. I think we need to be paying attention to these implications. About the same time, pheasant populations, ring-necked pheasant populations, had started a decline within this region. We've been seeing this for quite a few years as agriculture has become more and more industrialized and land use change has accompanied that. So we see these declines within pheasant populations. And then around 2008, there ended up being that little red circle decline right there. These population data are from Iowa. The folks who came up with the graph said, well, there was a lot of snowfall that year, so that must be what killed those pheasants. But it also happens to coincide with the year that neonicotinoids hit the market as seed treatments. And so we ended up getting a graduate student who decided to start studying this. This data has never been presented before, and there was a lot of efforts to try to make sure that you never saw this data. But the paper is now submitted, and so we're going to try to get this thing published and out to the world. Right. I left the names off until it's published because I don't want those scientists to be harassed before this even comes out. So not trying to steal any credit or anything like that, but it'll all be revealed to the world as soon as we can. Yeah. So what the grad student did was we took over the state fairgrounds and the poultry house out there, and we started to administer clothianidin, which is another neonicotinoid, treated corn seeds to ringneck pheasants under captive conditions to see what their how toxic these seeds might be to the pheasants. This is a survivorship graph, okay? And I think that so what you're seeing, it's a dose response, essentially. 75 treated seeds versus all the way down to 0 treated seeds. Okay. What I'm trying to show you in this graph is that the lines are all really overlapping a lot. Yeah, you see a little breakaway on the 75 treat, the highest dose there at the end, but essentially survival statistically was unaffected by the clothianidin treated seeds. What that means is that under this test scenario, under this hazard scenario, acute toxicity in the form of mortality does not appear to be having a direct effect on ringneck pheasant adults. Okay. However, the graduate student had the resourcefulness in order to start assessing sublethal effects, which oftentimes are never considered within risk assessments. What he found was that pheasants that were fed higher doses of that clothianidin ended up having more neurological behavioral disorders. They would twitch, right? They would be lethargic like the white-tailed deer were. So these would have really important implications for winter survival and predation rates on these birds under natural conditions. He also showed that the more that the pheasants were fed, the fewer eggs were laid. Okay, so these birds that were fed higher doses of imidacloprid and clothianidin were reproducing less. Not only that, but the hatch rates were also reduced. So it wasn't just fewer eggs, but it was also fewer chicks per female. Okay. And then he went so far as to monitor the survival of those chicks for 2 weeks after they hatched. These chicks had never seen before a molecule of clothianidin. However, there were significant reductions in their survival. Okay. So, so these sublethal effects, when you merge all of those sublethal effects together, what it means is that those pheasants, if a female consumes 5 seeds per day for 2 weeks, it would functionally remove 50% of the pheasant population. Boy, that's a huge whack at those pheasants. Okay, that's the hazard scenario, right? When on earth would a pheasant be eating those seeds under natural conditions? Well, um, together with the South Dakota Game Fish and Parks Department, they collected a whole bunch of birds and measured how much or how often we would find neonicotinoids within those hunter-killed animals. But first, Michael, I'm sorry, the graduate student ended up also testing the how or whether or not these birds would even consume neonicotinoids, right? Whether they would even be exposed to begin with. Behaviorally, if they don't like them, they're never going to eat them in the wild, right? And so what he found was that the birds could detect and avoided all seed treatments under these conditions. Okay, so within the laboratory, when given free choice, within 3 days, the birds had abandoned eating any dyed seeds, which so initially it feels like they were detecting, okay, the dye seems to be associated with a poor flavor or some health effects or something. And within 3 days, they avoided them. Then, after a week of this, or about 6 days, you start to see um, that they're able to actually distinguish between the dyed seeds that were untreated and the treated dyed seeds. So they could determine it's not the dye, it's the actual insecticide that we're trying to avoid. Well, that's great news, right? We shouldn't be seeing any implications then. They shouldn't be eating these things. They don't even like them. 95% of the pheasants under field conditions had consumed neonicotinoids within probably 24 hours of collection. Almost all of them. Where were they getting them from? And this was at a really pivotal time in the pheasant's population biology. They needed those seeds to develop chicks. And so the fact that they were all hot, yeah, has important implications. Where are they coming from? Other studies in Minnesota and elsewhere around the world are finding that these seed spills are incredible resources for a lot of wildlife, and pheasants happens to be one of them. They're consuming between 4 and 12 seeds at each visit, and they return to the same place multiple times in order to fully exploit these things. So our risk scenario that predicted 5 seeds a day for 14 days would kill a lot of pheasants. That seems like it's actually occurring under realistic conditions. Or at least it's one of it seems like temporally as well as from the risk scenario, it seems like this is probably has a lot to do with why we've seen that big dip in pheasant populations since 2008. So are they accumulating in food webs, right? DDT used to accumulate in these top predators. Those predators never actually consumed DDT directly. They consumed it via their prey. So North Dakota once again sent us some predators. River otters. Look at that cute little guy. Yeah, he's not going to do so well. You're not going to like this study either. So these neonics are they actually finding their ways into some of these predators? Okay. Bobcats, river otters, and fishers. So we tested them here at the laboratory. And this work was recently published. Um, yeah, I should say Elisa's deer study ended up in a couple of months, ended up going to the top 1% of all science ever written in terms of social media impact. So pretty damn, pretty damn good for a master's project. Um, Tremendous impact. This one was just released. We're awaiting final pagination on this one. What we found was that the fishers, the bobcats, and the river otters all were contaminated. 30% of those little cute otters ended up having neonics in their spleens. Where on earth are they getting it? These animals have never directly consumed a seed, right? They probably have very seldom, if ever, even consumed a plant. Where are they getting it? They're getting it from the water, probably. We know that surface waters and even groundwater are broadly contaminated with neonics at this point at meaningful levels. But the question is are they getting it from prey? Is it accumulating within these top predators, just like those early organochlorines and organophosphates? How am I doing on time? All right, let's see here. So that is, yes. How how did we get here? How on earth are we here again? Right? There's IPM was supposed to save us. Risk assessments are supposed to save us, right? The scientific community is actively engaged in this. Let's fold in one more layer that really adds to the complexity of this situation. These agrochemical companies have a, they have a they have a they will benefit directly from obscuring science, right? They yes. So science, there has never been a perfect study. All right. That doesn't mean we don't have to try, but but the reality is that science always has to be contextualized. Every single paper that's ever been written is not perfect. There's problems with it. There's strengths and there's weaknesses. That doesn't mean that we throw it out. That does mean that we have to think about the context under which those interpretations need to be made. And so science is not black and white. God, I wish it was, right? That's math, right? Biology is a much different branch of science. It tends to be more gray than black and white. It provides an understanding. It provides a framework for us to understand and interface with the world around us. And once you know that, there's a lot of ways that you can manipulate that science. If you have an agenda or you have a product that you're trying to preserve the life of and capitalize financially on, well, the process of scientific manipulation has been formalized. It was, this is for the tobacco industry is really where it came about. And if you ever want to see movies about it, there's good ones on Netflix or about Merchants of Doubt is a good one. The Insider, we just watched that one again recently. That kind of haunts my memory a lot. Um, Both excellent treatments of this. And if you really want to dig into it, the Merchants of Doubt is a really thick book on how science is manipulated by large corporations, written by Naomi Oreskes at Harvard. Um, yeah. So, whoa, what just happened there? I'm gonna go back. Jeez Louise. All right, there we go. That was it. That's a sensitive mouse pad. Woo! All right. This is heavy stuff. So, yeah, it gets better. Yes, we have good solutions. Okay. But I'm still going to bring you down even lower. So first, how if you have an agenda or you have a way or you have something that you're yeah, and there's a study that comes out and says that say your neonicotinoid is killing the bees. Good gosh. All right. Well, thank goodness there's never been a perfect study that's ever been conducted. Science is gray, it's not black and white. So, what you do is you go in and you figure out, okay, what are all the problems with that science? All of those studies that said the neonics are killing the bees, boy, look at it. It was done under this wrong scenario, and the bees were, in this state, and this does not transfer well to how on earth can we assess the risk of that? And so for that reason, it suddenly that piece of work looks like junk science and it can be dismissed, right? It's not hard to do. I mean, the scientific community is professionally trained to do this. Um, okay, so if science is gray, it's not black and white, how do we make decisions based on science? Well, you repeat the same study or similar studies under a lot of different conditions, and you wind up with hopefully what becomes a consilience of evidence, a preponderance of evidence that you can base a relatively secure decision on. All right. So, how do you get that? Boy, that sounds like a lot of science that has to happen. You pay for it. It's expensive. Who has the deepest pockets in the room? It ain't the little nonprofits that are working in a pole shed in the middle of nowhere, South Dakota. Okay. And so for that reason, okay, I got neonicotinoids are killing the bees. I know it. They are still unsure about it. I'm going to make sure that they don't come up with that conclusion again. We're going to fund every single varroa mite study that I can think of, as well as anything about flowers. Oh, and there's a new virus. Thank God for that. Let's give that money. And you don't fund any of the pesticide work. And suddenly it looks like the varroa mite is what's killing the bees. Okay. But then you have a noisy researcher that just won't shut their mouths. What do you do? You destroy that person, right? You rip them to shreds. You destroy their family. You go after not just them, you go after every person that they care about in their lives. And then you put their corpse up and you display it as widely as you can. And no scientist ever asks that same question again. That's science. That's how this happens. Yeah, so there I am. I'm had more hair, good looking gent, right? Met the president in the White House. Everything's going right in the career here. I've got a puppy mill full of postdocs and graduate students. There's one of my puppies now. We got publications out the wazoo in every peer-reviewed journal, top line stuff. Grants, I got millions. Everything that I was supposed to be doing, I was an advisor to federal governments around the planet, right? Everything I was supposed to be doing. According to the metrics of success and all of the stats that said you were a good scientist, there were very few people that had better stats than I do. To this day, I'm in the top 1% of all scientists in terms of research impact. But, and I met the president in the White House. He gave me an award for being one of the top young scientists in the country. Uh, but we started to meet beekeepers, and they were telling us the neonics are killing our bees. And and I explained to them, no, that's not true, right? The research isn't black and white on that. The research is gray. Yeah, maybe they're a contributor, but it doesn't seem like it's the smoking gun. And they told me, come on out and watch the bees die. And then you tell me that neonic on that corn seed during planting isn't what's killing my bees. And we started to do the science where we were connected to the problem. And the beekeepers were right. The neonics were killing their bees. So that didn't go so well. And then the farmers were starting to come to me, and they were saying, we're growing all of these crops and all of these livestock species, and we're not using any pesticides. They weren't organic. They had no ideology that they were really getting behind, right? They were just trying to save money. And I'm like, no, that's not possible. The science says that you are dependent on these products. You have to use them. And they said, well, come on out and take a look. And we did. And the farmers were right. They didn't need to be using all of these pesticides. And so we started to talk about that, and everything changed. Everything in life changed. No longer were we the golden children that could walk into any USDA office and or even the White House and talk about our research anymore. We were gagged. I had to pull my name off of science that was peer reviewed because it didn't adhere to the political agenda of the day. My staff was persecuted and interrogated like under a light bulb in the middle of a room, for crying out loud, to find anything that I might have done so that they could use it against me. Um, yeah, it really, it makes you ask the question of what the heck you're doing, right? I mean, I was following the rules. I was doing everything that I was supposed to be doing. And this was the response, right? Um, so yeah, I really had to ask the question of what is it? Why am I here? What am I doing this for? And the answer is that, I got kids at home. Uh, they're sick in ways that they shouldn't be. And um, and I want to leave the world a better place for them than what I'm getting. Um, and science is supposed to be able to help in that process, but it's become so corrupted the way it's been being done right now that it's really hard to know what's real and what's not. And so yeah, I quit. I quit my job. I quit Noisy. Um, Front page of the Washington Post. And I called it out. I blew the whistle. And yeah, everything in life at that point had fallen apart. My family was gone. I was on a intoxicated farm in the middle of nowhere, South Dakota, trying to bring something new together. Um, yeah, so that's that. I think this is something that we all could probably kind of adhere to. Wendell Berry is a real voice for a lot of really good things in agriculture right now. Um, Practicing resurrection has become a mantra for us, especially at Igdices Foundation and Blue Dasher Farm. Yeah, we're on the front lines of something here, and we're getting the crap kicked out of us pretty often, actually, for trying to do the right thing. And that doesn't make it easy, but you got to pick yourself up again, and you got to get back in the ring. So yeah, as a result of practicing resurrection, we have come to the realization that there's a better way. So, if we want to reduce pesticide risks, if we want to reduce the use of these things, there's two approaches. The approach that we have been going on, and I was one of the world's leaders on, was let's take a look at these individual chemistries, let's confront them, let's do the risk assessments, let's understand their action thresholds and restrict their use and things like this. IPM and risk assessment. I think that we can say pretty definitively, after 50 years of trying to use these approaches, Pesticide use is higher than it has ever been in human history right now. So it really begs the question, does IPM work? I don't think it does. I don't think it does. Does risk assessment work? I mean, scientifically, fundamentally, sure, these processes should be there. But when agro industry has figured out a way of manipulating this system so completely that they are rendered completely useless at this point, and we are being piled on with more and more chemistry. Okay, so that's that path. I tried to make it work. What's the other one? Change the system. Change the system. Because if we're expecting, or agro industry wants you to pursue lane one, they're ready for you. They got to the top of the heap by playing that game, right? By beating away any competitors, consuming them, by suppressing any science that might be obscuring it. They have figured that system out. If you change the system to one where pesticides aren't needed anymore, they don't have an answer for that one. They don't have an answer for that. So regenerative agriculture is where I pushed all of my chips onto. Thank God I did. Because the central problem that's influencing, my God, I'm talking for a long time. I'm almost done, everybody. We're just going to wrap up on a hopeful note. All right. This central problem that has produced this scenario where pesticides have become an addiction is that agriculture has become far too simplified. What does that look like? This is the percent of the total continental land use area of the United States. 5% is planted to corn, 4.5% soybeans, 2.5% is wheat, 3% is alfalfa. You know what? All of these are treated with neonicotinoids. 100% of them almost. Okay? That means 15% of our country is planted with 4 plant species that are all treated with pesticides and maintained with glyphosate and all these other chemicals that kind of change the whole risk scenario even further. What does that look like? Woof. Yellow is corn, green is soybeans. When the side of the jug says, you better ask your beekeeper to move his bees or her bees prior to you spraying this out there, the beekeeper that lives in this situation says, where the hell am I supposed to move them to? Right? I mean, you can see cities are the only things that aren't corn and soybeans, the gray areas in some of these regions. Is that insane? That is insane. We asked the croppers why does this matter? Why does this simplification matter? We asked the croppers whether they'd been experiencing any human health issues. 41% of them had allergies. 32%, a third of them have anxiety. A quarter of them have clinical depression. And then we compared those numbers with the general society, not just croppers, all people. Our croppers have 3 times the depression rate. They have double the asthma. And then we asked them about Parkinson's. And we asked them about cancer. We're literally killing our farmers. People that live within agroecosystems are experiencing health problems the likes of which we have never seen. Why should you care? Your kids, your grandkids are being affected by this right now. And unless we change the system, knocking out one pesticide at a time isn't going to move the needle. So what does a better way look like? I could go on and on about this topic. In fact, I have. There's other presentations. Maybe there'll be another webinar series. I don't know. Regenerative agriculture seems to be gaining attention on a national as well as a global scale. And we could talk about what that is at a different time for those that are interested. Or if there is a lot of interest, I have a whole bunch more slides that I could show everybody. But I'm not going to do that. I'm going to let people off of the hook here momentarily. So regenerative ag is starting to gain attention. And the question is, what are some of the implications of it? We've studied farmers that were transitioning large row crops, which traditionally would be treated with neonicotinoids. And what we're finding is that over a 3-year period, by year 3, 90% of these farms have completely eliminated all insecticides and fungicides. 2/3 of these farms have eliminated all herbicides and synthetic fertilizers. That's incredible. You think that they would get that information if they went down to the local co-op and asked the seed dealer what they should be putting on their field this year? Hell no. The only way that you're going to get this information is by independent science trying to encounter these things and measure them. So, yeah, why on earth would you be worried about herbicides and fungicides? This is something from Dan Pachenka's recent thesis. He looked at the relative effects of all of these different practices on invertebrate abundance in wheat in North America. And what he found was that, yeah, insecticides, if you want to kill a bug, use an insecticide. But if you really want to kill an insect, use a fertilizer. Spray an herbicide. That wasn't supposed to happen. That wasn't supposed to happen. Because regenerative agriculture fundamentally increases biodiversity. It is at its core. That is important. Okay. What we're finding is that, and this is just rangelands. I could show you the same graph for croplands, but I'm not gonna. And what we did is we went and measured. This is one of the largest bioinventories that humans have ever conducted. We've measured species on these different farms, and we've found an increase in every single organismal group that we have measured. And that life is how you combat pests. That life is how you restore ecosystem processes so that you are not dependent on these agrochemicals anymore. All of these other outcomes, as you abandon pesticides, all of these other outcomes that a regenerative system starts to produce becomes apparent. Carbon capture and storage, restoring water cycling, increasing the nutrition of food, improving farmer wellness and both mental and physical wellness, increasing the profitability and reducing pollution. All of that is an outcome of growing life on farms by reducing or eliminating entirely all of your agrochemical use. So, how do we break away from neonics? Step 1, stop using them. I mean, that's the easiest thing right there. Research shows that this isn't really helping. You guys saw that in Mike's presentation. Number 2, ask the question every time you are making a management decision on your farm or ranch or bee operation, is this action I am about to take growing life or reducing life on my farm? Number 3, stop disturbance. That means not punctuated disturbance. Yes, we don't want late successional communities. We want vibrant, actively evolving biological communities on your farm. That means punctuated disturbance is fine. Things like tillage or spraying everything out on your farm, those don't work. Those don't work. Diversify your operation. Don't bet the whole farm on corn and beans. That's dumb. That's a bad business decision. You got to be diversifying your operations. And then finally, start thinking locally. Start thinking within your community. This concept of industrialization and globalization of our food is not working. It is not working for the farmers, and it's not working for society. And I am going to shut up now. If anybody would like to know, yes. So, yes, these are some of the folks that have donated to make all of our research free and available. And if you're interested in pursuing 1,000 farms and regenerative agriculture, register your farm here. We might be able to help you. And with that, I'm sorry I spoke for so darn long, but I had things to say. I had things to say.
That was
All
great,
right.
John.
What do we got, Ryan? Turn it back.
Yeah, if we got a few minutes for questions, we had a few roll in on the Q&A here. I'll just start from the top here. One from the biopharmaceutical manufacturer, and they're relying on bees for making life-saving vaccines. They ask, is anyone gathering data on the impacts of pesticides on protein and enzyme activity in pollinators?
Jeez Louise, I'd have to scan the lit for that one just to know whether, but I don't know of any research groups that are examining that at this point. There might be some disruption to particular hormone cascades or something like that that could be probably dug out, but yeah, that's a pretty sophisticated question that I don't have at the tip of my tongue.
Yeah, understood. Oh, and just so folks know, the we did record this webinar and it will be posted to Igdysis's YouTube channel, just to clear up that question. We had one about talk about the ways chemical companies try to silence research. I think you did that pretty
Yeah.
well, unless you want to add any more to it, John.
No, I think I said all I have to say about that.
Okay, okay. Here's a good one. They ask, so many conservation areas have corn, sunflower, etc. plantings for wildlife.
Hmm.
Do you know if there's any management planning with regard to treated versus untreated GMO seeds in those areas?
Most of the GMO seeds that I know of are treated. You have to go fully conventional or naked seed in order to and there's a lot of companies that are selling that. It may not be the local co-op or your brother-in-law, but you can find them. There's a thing called the internet, and you have to Google that one, but it comes up with quite a few options for you if you're interested.
Yeah, yeah. Oh, here's a good one. How do we get agronomists on the right side of this conversation? As many farmers do whatever their agronomist says. Also, the insurance and banks that say you have to treat for crop insurance.
Yeah.
That's yeah,
Crop insurance is one of the worst things for farmers. It is stifling the innovation of our food system, and it's going to be really hard to move the needle until we dramatically reform crop insurance. And I'm not saying don't have a safety net for real emergencies, but when crop insurance and subsidies are supporting a simplified system and mandating that they apply some of these things, you know, get off of the hush money, folks. Why on earth are you dependent on these federal funds? This is welfare for farmers. Um, yep, time to change it. Grow something else. How about that? If you diversify your operation, the diversity of products that you end up supporting or producing is the only farm insurance that you should need.
hmm, said. Um, oh, we did have a follow-up as to um, the protein content
Oh, really?
in the pharmaceutical. They said I just put this out there
For
for folks.
context, yeah. What? Yeah.
Specifically, we are testing to we are seeing higher protein content and hydro oh boy, I need help with that word. Hydroluridase activity in our bee and vespid venom, and we have a hunch that it may be related to pesticide use, but we can't prove it. So yeah, that's
All right. Well, that'll be interesting. Let's yeah, see where that one goes.
awesome. We got another question roll in here. Are there any studies that look at neonic accumulation in feedlot finished beef versus grass finished beef slash grass fed slash grass finished beef?
Not that I've ever seen. I don't believe it exists. I think I'd have come across that one. And it's one of those things that we would be love to be studying some of this aspect of environmental contaminants within the Thousand Farms Initiative. But we've never had the funds in order to be able to do that. So there's some real opportunity for digging in even deeper within some of these beef samples and other sort of yield products that we could explore in the future.
Yeah, yeah. Let's see. I was, I guess we had another question of in your deer study, we saw that all those related effects between neonics you found in the spleen and correlation to, those deformity outcomes and health outcomes. Could you expand a little bit more on why,
Hmm.
neonics in the spleen potentially causes? I know you're not a physiologist, but what do the spleens do, and how might they lead to those outcomes?
The bottom line is that we don't understand the complexity of the mechanisms whereby pesticides affect higher organisms very well. I mean, we can make some assertions as far as things like the disruption of particular neurological functions or something like that, because it is a neurotoxin. Yeah, we have a different acetylcholinesterase receptor site, at least it's structurally just slightly different than insects have, but that doesn't mean that it's not affecting us. And there's going to be a lot of different ways of doing that. Tripping hormone cascades, I think, is going to be one of the more important ways that these pesticides are going to affect some of our higher organisms like mammals and birds and things like that.
Mhm.
But good question, and one that could support a whole career's worth of research.
Hmm. Yeah, for sure.
Really important.
Here is one from our friends in Florida. They're asking, how can we help our university researchers do the studies that show the truth about regen ag as a system and its benefits? Shockingly, they are asking, they state.
That's great. Let's see. Farmers get organized. I would think that would be really important and give them some best management practices. We do not want them doing research on experiment farms. We want them to be doing science and ecosystem monitoring on operating regenerative farms. The partnership that used to exist between scientists and farmers really got disconnected as the scientific community became more focused on alternative metrics of success from what the farmers really needed or were interested in. So, um, yeah, I think that the scientific community is going through a real evolution right now, and searching their souls a little bit about what this is all about, because there has been funding restrictions and there has been a lot of ethical and breaches, and honestly, it's getting harder and harder to attract the next generation of scientists into that field, at least at the university level. And so I think that we have to do a little bit of soul searching to figure out what is it that we're really asking. If universities' business models are completely dependent on outside grant funds for in order to keep their doors open, and the only way to do that is by using these patsy scientists to write grants left and right and think that they're achieving something grand by doing so. Uh, yep, they drank the Kool-Aid in the wrong direction. And listen, I was a part of that world. I was good at it. It wasn't until I got out that I saw the light.
Thank you. We had one come in from South Dakota, a local one here.
Nice.
They say, I think we can assume that similar results found in pheasants would be seen in waterfowl produced uh, in the prairie pothole region. Any thoughts on that?
Yeah, we have a multi-year data set looking at ephemeral wetlands that are actually in croplands, and we've got some population level for waterfowl because this is a major migratory route for a lot of waterfowl. I think the physiological effects are going to be very similar. We have not done the analysis on that yet, so I can't really speak too high or too broadly on that one, but I can tell you that the surface waters are they have crazy high amounts of neonics in them.
Yeah. Um, oh, maybe one last. How much time are we doing on time here? We had one more roll in. I guess if people keep firing them and want to stick around, we can keep going. We had a question. Can you talk about the way agribusiness is patenting biological solutions rather than just accepting a paradigm shift? It's a good one.
Patenting biological solutions versus adopting a functional ecosystem.
Hmm.
The farmers, if regenerative farmers, if all they're doing is just buying the next regenerative product or replacing their conventional products with regenerative products, they're dead in the water before they even get started. And that's not to say that those might not be helpful in getting things going on their farm, but you have to have a functional ecosystem in order for a lot of those products to even work. And their best tools at the end of the day are going to be plants and animals. And those are real tough for large corporations to patent. So I think that the only way that, listen, under no scenario within the current system, do conventional farmers win? Right? Anytime that they make an extra buck, the seed salesman, the chemical salesman, the implement salesman are all poised to squeeze every last drop of new money out of that farmer. They do not win. The only way that they win is by changing the system, by getting out from underneath that. And yeah, I get it. It's scary. Nobody knows that better than I do. All right? I was pretty I was pretty comfortable in my federal position, but it wasn't doing what it needed to do.
Hmm. Yeah. Uh, we had another one come in from Regen Ireland.
Hey,
In your experience, when all inputs are eliminated when practicing regen ag, what is the first bio indicator that improves?
life returns to your farm. You so as a farmer, what's the first thing that you see? Probably insects, probably birds. If you have the right technology, you could probably measure microbial shifts, plant communities resurging on your farms. And that's been something we've seen in darn near every system that we've examined so far is that returning of biodiversity is this linchpin that ends up supporting so many of the other ecosystem functions. So, carbon accrual and storage and poking holes in the ground to let water go in and regulating those water cycles. All of it are artifacts of having additional life on the farm.
Nice. Thank you. Well, unless there's any other comments from the folks in the room there at the lab,
Yeah. Anybody
we've
have any questions? It's kind of a long time. They just woke up and a little bit of it's all right. Okay.
okay, wonderful. Well, thank you again, John, for putting that together for us. That was a great wrap up to this series. Again, for people that are looking for this, these will be posted to Ecdysis's YouTube channel, and you'll be able to see Dr. Bredesen and Dr. Wu Smart's talks there as well. Um, And yeah, I guess do we have one more coming up, not on neonics, but we have another webinar before the summer kind of hits.
Oh, on birds?
Yep, on birds. So if anybody that's on this one, if you want to come and we'll have our ornithologist at the lab discussing birds and regenerative ag, and we'll let them decide on the specific topic. So that'll be kind of mid to end of May. So look for that coming out soon too. So thanks everyone for a great series here so far.